Talk about the cell cycle. The cell cycle has interphase and mitosis. What consists of interphase?
G1, G2.
G1. You have also M phase. You have an S phase, and you have a G2. Do we have a G6? So the G1, what is that? What point is that?
Replication.
It's a like when you're passing the border, there's a checkpoint. There's a checkpoint. Oh, okay. Checkpoint, and then G2 is another checkpoint. The S phase, that's what happens? That's semi-conservative. Replication. So we're a replication of the DNA, right?
Right.
And G1, S, and G2 collectively, that's interphase. Interphase is the majority of the cell cycle. Okay? And then we have M, which stands for what? Mitosis. Right. And how many phases in mitosis are there? Four. So I remember PMAP. Please make a tortilla. I used to date this Hispanic chick. Her mom cooked a lot, and I'm like, please make a tortilla. And then I'm going to describe to you what happens in prophase. In prophase, there's things that disappear, and there's things that appear. One of the things that disappear, what do you think it is? The nucleus. The nuclear envelope starts to disappear. So instead of drawing a solid line like I did previously for other diagrams, I brought a dotted line. Okay. And what appears? Basically, it's the chromosomes. And also the spindle fibers. And the spindle fibers are attached to centrioles, right? So let's go back to chromosomes. This is a sister chromatid. Two sister chromatids together make a chromosome. The part where it's in the middle is the centromere. Okay? Good. So you could either write down, since you're beginner level, I would write down and also draw this picture. Okay? So what happens in prophase to the chromosome? Is it condensing or decondensing? Condensing. So another word for condensing means that it appears. What happens to the nuclear envelope? Disappears or appears? Disappears. What happens to spindle fibers? Appears. And centrioles? Appear. Cool. So that's prophase. Telophase is easy because it's opposite of this. Total opposite. And in telophase, basically your nuclear envelope comes back, right? And you have two of them. What happens to your spindle fibers? That's why I didn't draw it, because they disappeared. What about the centrioles? That's why I didn't draw it, because it disappeared. What about the chromosome? Decondense or condense?
Decondense.
Which means disappear. Disappear starts with a D. Discondense starts with a D. So kind of the same. You could link it that way. Okay. And then we also have an additional thing. Cleavage. Do we find cleavage in plants or animals? Animals. What about in plants? It's a cell wall.
Reproduction cell plate.
I had more homework on cell wall. Who's?
It's 100% it's in the notes.
I'm going to look that up.
It's a cleavage furrow in animals and
Yeah,
a
I know it's
cell
cleavage.
plate
Plates.
in the plants.
Plate in plants.
Yeah.
It
Cleavage
goes from the inside
animals.
to the outside.
Yeah, because I have this. That's my cleavage. And then plants. How can I read? Plant starts with a P. Plate starts with a P. Plant's plate. Good. I got that. Thanks for teaching me. So in metaphase, you're gonna think metaphase plate, and that's when basically it lines up in the middle. M starts with an M, right? I mean, M starts with an M. Middle starts with an M. Middle starts with an M. So imaginary line. You have your centrioles attached to the centrosome of the sister chromatid or the chromosomes, and that's what happens. So if I say keyword plate, what phase are you thinking about?
Metaphase.
Metaphase. So anaphase, this is when sister chromatids separate, right? So I'm still going to draw the middle to show a point of reference. But the centrioles are pulling the sister chromatids to them, like tug of war, or like a sailor getting the anchor from the boat. That's what's happening here. Okay. So that's important. Kaplan wants you to know that. And telophase, opposite of prophase. We have in animals and a plate where? In plants. Okay, good. So mitosis is like a copy machine. You're going to make more of the same cell. So in mitosis, you start off with something that's 2N. If it's a copy machine, what are you going to make? 2N. Right? And you're going to have two of them, like you have here, when it's done, 2N. So some things, or some type of cells that can regenerate, continuously regenerate. What regenerates? Skin. Sperm cells, yes. Also, skin, epidermis. So when the FYI, when your epidermis sloughs off, that's actually the dust in your house. Also, I had a crazy night, drank lots of alcohol. I effed up my liver. The liver regenerates, but only to a certain point. Like, you can't drink 40s like 3 times a day and it's not going to regenerate. Or a 4 Loko. 4 Loko is crazy. I'm just kidding. I'm being recorded. Edit. Do you do editing in addition to filming? No.
I'll get into the computer a little bit, but...
Okay, so meiosis. It's not a copy machine, it's a sex machine. Come on, guys. Sex machine. And in meiosis, do we have 1 or 2 divisions?
2
2. We have meiosis 1 and meiosis 2. I'll save you a little bit of work with meiosis 2. Meiosis 2 is exactly like mitosis. Good. So in meiosis, you basically start off with something that's 2N. After meiosis 1, you get 2N and 2N. But since you're making sex cells, there's going to be haploid. Hi. Oh, you got one of these too?
Yeah. You're starving.
And you're going to make N, N, N. 4 N's. N, 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 N. And haploid or 1N are like the sex cells. Sperm or eggs. Okay, so that's a sex machine. Prophase 1.
Len?
Yeah.
Sorry,
Yes.
for the first division,
Yes.
is it supposed to be 2N to become N, and then it becomes...
Let me double check.
This meiosis 2 is the same as mitosis, so it should go from N.
Yeah, you're right, actually. It's N. And with prophase 1, the nuclear envelope should disappear. Centriole should. Spindle fiber should appear. But are we dealing with homologous pairs or sister chromatids? Homologous pairs. So they're in pairs, kind of. And I drew 2 pairs. And That's a blown up version of my nucleus. Okay, and you told me that the nuclear envelope disappears, so I have to draw dashed lines because that signifies. Also, you told me centrioles appear, I draw those, and they're attached to the homologous pairs, as so. And in prophase 1, there's a crossing over of tetrads. Why do we call it tetrads? Because we have 4 sister chromatids per. Let's just look at this one. 1, 2, 3, 4. That's a tetrad. Okay. So crossing over, it's very random. You switch parts. Like Transformers, Autobots, and Decepticons, they're like trading parts with one another to generate a new robot.
So we need to know the tetrad, but you don't need to know like the bivalent or like zygotine or the.
Beyond scope.
Yeah.
Where did that come from? From class?
Yeah.
Old classes? Yeah, beyond scope. So that's pretty much it in prophase 1. And then in metaphase, I kind of set this up for metaphase already, but it's going to line up on what?
Metaphase
Plate, yeah. Metaphase plate. Is this metaphase plate the same as in plants? No, that's like for the division, opposite of like, or related to cleavage. Please make a tortilla. What does A stand for?
Anaphase.
Anaphase. So in anaphase, what happens is chromosomes randomly go to one side. Instead of sister chromatids, I'm just replacing the word sister chromatids with chromosomes. When they were together, they were homologous pairs. And when sister chromatids together, they were chromosomes. Right? So now they're individual chromosomes migrating to either the left side or right side, and there's a 50-50 chance of that. So that's another source of genetic diversity. What was the first source of genetic diversity that we covered in prophase 1? The crossing over. The switching of like different parts of the chromosome pairs. So this could be like 1 and 2, this could be 3 and 4. That's one possibility. Another possibility could be, so 1 and 3 are actually on this side, and 2 and 4 are on that side. Another possibility could have been 1 and 2 and 3 and 4. 1 and 2 on this side, 3 and 4 on that side. So it's an, yeah, there's law of independent assortment, which is Mendel's second law. And we're going to cover that as well. Okay, that's towards the end of our class. Okay, so any questions with meiosis? Your choices are mitosis or meiosis. Copy machine.
Mitosis.
Sex machine.
Meiosis.
Haploid cells made.
Meiosis.
One cell division. Two cell divisions. Crossing over. A little more specific on that last one. Prophase 1, not prophase, there's no prophase 2 for crossing over. Sources of genetic diversity, prophase and anaphase, which is found in mitosis and meiosis. Okay. In mitosis, I have 2N. I'm going to make two 2Ns. And in meiosis, I'm going to end up with 4, which are haploid or diploid?